Our guest today is Dr. Jessica Grembi. Dr. Grembi is an assistant professor in the Department of Veterinary and Biomedical Sciences at Penn State University. Her current research focuses on host microbe interactions in the human small intestine and the role of microbial ecology and diarrheal disease in childhood stunting. Her lab is focused on understanding the mechanisms underlying environmental enteropathy, a subclinical condition which plays a role in persistent undernutrition and is estimated to impact between 50 to 90% of children in low and middle income countries. And with that, I will turn it over to Dr. Grenby.
Thank you all. I'm really excited to be here and share some of my past and current work. And as the title says, I'm going to talk about environmental enteropathy. But before I even tell you what that is, I'm going to assume most people haven't heard that. But I want to tell you a little bit about why I care about and kind of how I came to learn about environmental enteropathy. And that is because I started studying during my PhD childhood undernutrition. There are almost 150 million children globally that are stunted, and stunting is a form of chronic undernutrition. And that is roughly a quarter of the population of children in the world. And childhood undernutrition is multifactorial. Children are often, so just to kind of orient you to this plot, we measure stunting and childhood undernutrition by a child's height for age z-scores. So a healthy child is at the 0 z-score, kind of the average, the standard, and this is across global populations of children. And then we define stunting as 2 standard deviations below that norm. And so in many low and middle income countries, children are born at varying degrees of this signal of undernutrition. And that's primarily due to maternal undernutrition and in utero growth faltering. So you can see from these 6 different low and middle income countries that children, and these are population level averages for these countries. So certain countries, children on average are born kind of worse off than in other locations. However, we see this really, really drastic drop off between the age of 6 to 12 months, and growth velocity drastically reduces in children. And again, these are population level averages for these communities. And so this period right here of kind of 6 to 24 months is a period where a lot of different things are happening. First of all, exclusive breastfeeding is stopping and children are being introduced to complementary foods. Children are also becoming more mobile, so they are now exploring their environment. So they're actually exposed to a lot more things during this period of time. And this is also due to the change in diet is a very important period of microbiome development. So this is where we see kind of children from many, many different cultures and populations have this very similar microbiome when they're born, and they're exclusively consuming either breast milk or formula. And then as they begin to eat a diet more similar to their parents, their microbiome kind of matures into this more adult state that we see. So many people in the nutrition community, and I should say, these are locations where nutritional supplementation is often provided via government programs to try to reduce this kind of chronic stunting at the population level. But these interventions have failed to make substantial differences. So many people in the nutrition community believe now that children are actually unable to absorb the nutrients that are provided in these nutritional interventions. And that is because they believe that children have environmental enteropathy. And so what is environmental enteropathy? I'm showing you some figures here, which basically show changes to the villus architecture in the small intestine. So a typical small intestine has intestinal villi that are finger-like protrusions that have a lot of surface area available for nutrient absorption. And environmental enteropathy is a pathophysiology of the small intestine where these villi become blunted, so they shrink down and they kind of swell together. So substantially less surface area is available for nutrient absorption. And there are several different synonyms. Many people call this environmental enteric dysfunction. And there's also the term tropical enteropathy. And so these are all kind of synonyms for the same condition. And again, it's a pathophysiology, and it's really characterized by this intestinal permeability and intestinal inflammation. So the inflammation obviously reduces nutrient uptake, but the permeability also allows for microbial products and microbes themselves to access kind of the inner layers of the human gut, so they can basically stimulate the immune system much more, which leads to kind of this cycle that the immune system becomes, I would say, hyperactive, or reacting very normally. Hey, these things are not supposed to be here. But particularly for children, when they're growing, nutrients need to go towards growth. But when we have this constant systemic inflammation and systemic immune activation, that's actually partitioned as part of like normal body functions. And the body will support the normal body functions before it supports additional growth. And so really it becomes this vicious cycle that the body needs more nutrients for growth, but those nutrients continue to be diverted towards this immune activation at the same time that nutrient absorption is reduced by the condition. So it's a pretty complex condition, and it actually has quite a long history. And so I want to tell you a little bit about the term tropical sprue, which was actually first described. This condition was first described several centuries BC in traders to the Indian subcontinent. And it's really characterized by intestinal malabsorption and chronic diarrhea. And that kind of in the long term led to nutritional deficiencies. So anemia, vitamin B12 deficiency. However, this condition primarily was observed in adults and not in children. And it is kind of believed to have an infectious etiology because once antibiotics were discovered, it actually responds quite well to antibiotics. So again, this has been many centuries, this condition has been observed. But in the 1960s, people started describing actually a kind of a dampened version of this, which was called tropical enteropathy. And that's just minor abnormalities in the intestinal mucosa. This was observed both in adult and in pediatric populations. So kind of unlike the tropical sprue. And what's really interesting is in the 60s and 70s, we started noticing this in Peace Corps volunteers and Vietnam veterans returning to the US. So they had diarrhea episodes, but those were cleared up, but they had this long-term and major symptoms were, I'm very tired. And what their intestines were biopsied and they had this low-grade inflammation. And it actually cleared up without any treatment 6 to 8 months after repatriation back to the US. And the same thing was seen then in migrant populations moving from, let's say, the Indian subcontinent to the UK. So from tropical climates to a more temperate climate. And so around this time is where the term environmental enteropathy was introduced, because it was actually started to be observed not only in tropical regions, but also this idea that if you remove someone from the environment that they're in, then the symptoms go away and resolve on their own without the need for any actual other pharmacological intervention. So there's been, I would say, if you read any paper on EE, the primary cause will be that everybody talks about is just this chronic exposure to enteric pathogens. So I'm showing here the 5F diagram. So I actually came into this field because I'm an environmental engineer and I was working on these water and sanitation projects. And so the 5Fs are fingers, flies, fields, fluids, and feces. So an infected person, transmits via the fecal-oral route, because if you open defecate, if you don't have sufficient sanitation facilities, that fecal material will then be transmitted to another person via one of these other Fs, via hands, via flies, landing on the feces. Feces remain in the field and then get onto agricultural products. and then obviously it can contaminate drinking water. So the blue bars here represent kind of engineering controls and barriers that we can put in place to break down these pathways. So definitely we believe that enteric pathogens have some role in environmental enteropathy, but I'm not convinced that they are the only exposure and the only cause. And I'm not actually convinced, how, I wouldn't say like 50%, like to give a percentage to it, I'm not sure. So there are also, we know that poor nutrition actually can result in intestinal permeability. So if someone is on an undernourished diet, then they can develop the permeability, which can then induce this cascade of inflammation, which leads to more permeability, which leads to more inflammation. And so could diet alone, be responsible in particularly in children. But then there's also exposure to mycotoxins in staple crops. So maize and rice, especially in tropical areas, can be contaminated with fungus that is inherently not itself toxic and is actually killed during the cooking process. But the mycotoxins, like for example, aflatoxin is a big one. There was actually a study a few years ago in Nepal that showed many children had aflatoxin detected in their blood. And it was believed to be through this contamination of staple food products because the toxin is not deactivated or modified in any way during the cooking process. So even though the actual bacteria, the actual fungus is gone, the mycotoxin remains. There's also evidence that show that industrial chemicals can cause intestinal permeability and inflammation and irritation. And particularly in Bangladesh, where I work, you see people spraying. There's been some nice agricultural studies showing that in many places in Asia and Africa, pesticide use is, the concentrations found in fields are much, much higher than what is recommended by the manufacturers and then what are recommended as like safe levels. And so I think there is a disconnect between poorly educated users of these products to like what's safe. So typically no kind of PPE or anything is worn. So a father would be bringing those chemicals home on his clothing when he comes to the house at night and is holding his baby, as well as just the residual exposure from what is on the crops, what is getting into the drinking water, et cetera, that these children and adults might be exposed to. And then there's non-pathogenic microbes. So there are some nice studies from Jeff Gordon's lab showing using duodenal aspirates from the small intestine. He's generated microbial communities that can induce in mice this kind of chronic stunting. And so none of these are known pathogens. So just the idea that the microbiome itself could be in a state that extracts less nutrients from the food that it's provided. And in the case of someone living in the US, it might be, oh, that's actually a great phenotype because you can eat a lot and then you won't put on weight. But for someone whose diet is insufficient, that's actually could be leading to this chronic undernutrition. And then there's also the component of farm animals, particularly in rural areas where I work. A lot of people cohabitate with their animals. So either the animals live in the compound and also defecate in the compound, or even within the house. People in rural Bangladesh, store their chickens under the bed at night so that predators and or other people don't come and steal the chickens. And so if you imagine the chicken is under the bed at night, defecating when it needs to, and then the next morning, the child is now playing in that same space. So there's a lot of exposure to microbes of animal origin that I think could also be important and relevant in environmental enteropathy. So really, I believe, studying environmental enteropathy is a One Health endeavor, and definitely, it kind of ticks on many of the things that I'm sure you guys talk about kind of on a more regular basis. So really, it's a complex, multi-factor etiology and pathology. And so there are a lot of challenges to studying environmental enteropathy. The first being that there's really no precise clinical definition. So we go off of these intestinal biopsies and this change in the villus architecture. But biopsies are expensive, they are invasive, and they're not primarily done on a routine basis. And the facilities aren't available in a lot of the regions where we believe environmental enteropathy to be highly prevalent. So there are also, due to the inherent risks, so you can actually perforate the intestines during a biopsy accidentally, which could cause someone to go into sepsis and die. And so for that reason, it's very difficult to get IRB approval to get healthy controls. So now we are comparing people that have some indication of some GI issue that they need a biopsy or endoscopy anyway. And then we're comparing those to people that we suspect to have EED. So we don't really have good healthy controls in most of our studies due to these limitations. So this is all resulted in the use of biomarkers. And these are non-invasive biomarkers. We collect fecal samples and we study different measures of intestinal inflammation and permeability. We do a dual sugar test. So we basically give people 2 different sugars, a short and a long chain sugar. Then we collect their urine for 5 hours. So if anyone has a toddler or has been around a toddler, now imagine affixing a urine bag to their genitalia and telling them, now just play normally and I'm going to collect all the urine that you produce in the next 5 hours. So particularly in pediatric populations, this is really challenging. And we do it in the field, but it's not fun really for anyone involved. And we also do measure some systemic measures of inflammation and epithelial injury and microbial translocation in the blood. But the challenge with these biomarkers is that there's not a gold standard. This biomarker equals this histopathology. There have been a few studies that have tried to benchmark these biomarkers against histopathology. And in fact, we often find that there's zero correlation or very, very poor correlations. There's also poor correlations between the biomarkers themselves. So there's not a single gold standard. There's not even one that kind of tracks, and they often go in different directions. And so it's really muddled the field and made comparing across studies really difficult, and also made it really challenging to assess intervention effectiveness. Because which of the biomarkers would you count as a good intervention? If it changes one but not the others, was the intervention successful? And due to kind of all these limitations, really the true prevalence is unknown. We have no idea how many people this population affects. We can't do biopsies at the population level. And we use these measures, the absorption test, as kind of proxies for what the prevalence that would be, and we expect it to be high based on these studies. But also, there are clinical, like downstream clinical outcomes that we use. So undernutrition, so the malabsorption leads to undernutrition. There's also poor vaccine efficacy. Many vaccines that are trialed in the US or in Western populations actually show way worse efficacy in populations in low and middle income countries where those vaccines are actually most needed. And we believe that that environmental enteropathy is a big component of that, because the mucosal immune response is definitely altered. And then, of course, for children, as I started at the beginning telling you how important we believe this condition is for growth. And so the biomarkers sometimes track with child growth and kind of future growth faltering, but not always. So we really, I hope that I've kind of convinced you that EE is widespread, it's poorly defined, it's historically conflated with other conditions like tropical sprue, which are actually, I believe, quite different. And it's just really difficult to study because of the inaccessibility of the small intestine. We know it matters for child growth and nutrition, but we really lack kind of the epidemiological data on prevalence and really the means to assess kind of any kind of intervention performance. And so since the small intestine is the epicenter of EE, we really need some better way to study it. And I just want to also emphasize that even if we could biopsy everyone, these are invasive and costly, so it's not realistic and feasible to do at a large scale. You also can't access the entire small intestine with biopsies. So you can do endoscopy and go down and get the kind of the beginning of the small intestine, the duodenum, and maybe the beginning of the jejunum. But really the ileum is the largest part of the small intestine, and it's fairly inaccessible. Also, Endoscopies require sedation and fasting. So we don't get a true picture of what, let's say, the microbiome might look like during normal digestion, and what bile acid profiles might look like in people as they're actually digesting their nutrients. And so some would argue, yes, when you don't have a good human model, then you move to animal models. But actually, the digestive anatomy and physiology in animals is quite different than in humans. And the most common, animal model is mice. And the biochemistry is actually quite different. So just as one example, in humans, our bile acids are 75% glycine conjugated and about 25% taurine conjugated. In mice, 95% of their bile acids are taurine conjugated. And taurine contains a sulfur group, and so it's quite different biochemically. So I believe we should be studying humans because this condition affects humans, and we are a very unique species in and of our own. And so we need tools that we can use, and we now have those tools. So I'm going to tell you now about a new capsule sampling device that we've been using for the past few years. And I'm going to start by telling you about a study that we did that we published a few years ago in a healthy cohort of 15 individuals. And then I'm going to tell you a little bit more about how we're using that now to actually study environmental enteropathy. So in the first study, we have these capsules. And here's what they look like. They look like this little white pill, like a normal kind of Tylenol gel cap that you would swallow. And inside of that rolled up is a silicone bladder that when the capsule coating dissolves, that bladder unfurls and then can collect an aliquot of luminal fluid. And in fact, for this study, we had subjects swallow 4 different versions of this capsule designed to open at different areas of the small intestine. So some of them had a coating that was designed to open in the duodenum or jejunum, and then the coating was modified so that the later capsules were designed to open in later sections of the small intestine. And we actually asked subjects to swallow 4 of these capsules 2 hours after lunch, and then 4 more 2 hours after dinner, and then the next day to repeat. So our subjects swallowed 16 capsules over the course of 2 days. We retrieved all the capsules, no issues. And I'm going to just share a little bit with you today about the microbiome and the bile acid profiles that we did, just because I want to kind of talk about some of the new work as well. But this paper is already published, and you can find kind of all of the data there. And we actually have an additional paper really highlighting the bile acid and metabolomics piece as well. So the first thing is that the microbiome of the small intestine is actually quite distinct from that of stool. And so here, this is a PCOA, a principal components analysis, and each point represents a sample. And we had, of these 15 subjects, we collected saliva at the time that they swallowed the sample, at the time that they swallowed the capsule. And then the capsules we retrieve in the stool. So we also have an aliquot of stool from the bowel movement that the capsule was retrieved from. And so we see that the saliva is, very distinct microbial community. And basically the distance between points here represents a more dissimilar, so the larger the distance, the less similar the microbial, the overall community is. And so the saliva is quite distinct. And then on the far right side here, we have the stool, and then the CAP scans in the small intestine. And if you think about your GI tract, the input being the saliva and the output being the stool, as we would expect, the CAP scan kind of falls, along this spectrum. And we did have 2 subjects who are highlighted here in solid filled shapes. And those 2, one of the inclusion criteria for our study was no antibiotic use in the previous 2 months. These individuals had actually taken antibiotics 3 months previously and 5 months previously. So they were actually in both their microbiome and their metabolomics, we saw distinct differences, indicating to us that antibiotic use, those modifications to the microbiome and therefore the bile acid pool actually are sustained for longer than 2 months, at least in these study participants. We see that there are different taxa that are more abundant in the small intestine compared to the stool, and that is primarily Escherichia, so E. coli, and also Bellophila wadsworthia, which is a known bio-loving microorganism. We also observed that the microbiome was more variable within the capsules than a stool sample. So we know that within an individual, their stool samples might vary a little day to day. And that's what's shown in this brown box. So just to kind of orient you, the left side of this plot, the samples are more similar to each other. And in the right side, the samples, the microbial community as a whole is more distant. And so we're looking both within a subject, their stool samples are actually more similar to each other than their small intestine samples. So these 16 samples taken over the course of 2 days are more different than the stool samples taken over the course of those 2 days. And that's within a, yes, please. Okay, and we see across subjects even stronger differences on this kind of between individuals, and so the variability is high. It's a very dynamic environment in the small intestine, and probably that's because these are samples taken during digestion, like while people have just eaten, and there are blooms of certain things that are metabolizing whatever is in the gut in that moment. Unlike the large intestine, which is kind of I would say, a more constant environment. So we also know that the microbiome can modify bile acids. So it can actually deconjugate, so it can remove that glycine and taurine from the backbone of bile acids. And it can also dehydroxylate, which is the removal of this OH group to make primary bile acids into secondary bile acids. Now, the deconjugation primarily occurs in the small intestine, and the dehydroxylation primarily occurs in the large intestine, so in the colon. And so what we found in this cohort was that when you look the difference between the capsules and the stool, you actually see, so the primary bile acids are the ones in reds and yellows, and the secondary bile acids are the ones in the purple shades. And so you can see that the stool had a much greater percentage of these secondary bile acids, whereas the small intestine had a much larger fraction of the primary bile acids, as we would have expected to see. We also see deconjugation occurring throughout the small intestine. So the devices that were intended to open in the most proximal areas actually had the highest fraction of liver conjugated bile acids. So those are the ones with glycine and taurine. And as we progress down closer to the distal ileum, we see that fraction, that percent, goes down. And so we do see this deconjugation occurring. What's also really exciting was around the time that we were doing this study, there were other groups publishing that this deconjugation that microbes do in the human gut, there was actually hypothesized that microbes could then reconjugate different amino acids to that bile acid backbone. And so about 5 years ago, so right around the time our study was kicking off, they actually discovered this reconjugation with phenylalanine, leucine, and tyrosine. So kind of historically, for hundreds of years, we've known about the glycine and the taurine conjugated, and these three were discovered, and it was proven that microbes were the ones reconjugating those. And in this paper in 2021, there was this hypothesized, basically any amino acid could then be reconjugated to the bile acid backbone. And this is one figure from that paper. And we found all of these highlighted in green, circled in green, in our human subjects. And so it is this new frontier of human biology and of biochemistry that we've been unable to study in the past due to inaccessibility. And we're seeing these now in our human subjects, which is really exciting. And so now we have this new technology. We can now study the small intestine. So great, now let's use it to study environmental enteropathy. And so what I'm going to tell you about next is a study that we have completed and we're currently ongoing with the data analysis, where we actually use the capsule device in pregnant women that were being given interventions to improve their gut health. And this is work funded by the Gates Foundation, and it is a series of intervention studies, and we are currently in the field rolling out the second intervention study, but I'm going to tell you about the EMP1 study right now. And that is across 4 different country sites: Bangladesh, Pakistan, Zambia, and Senegal. And we enrolled, the larger study had double these numbers, but we gave CAPScan to about half the women at each study site. So 30 women from most of the sites, except for Senegal, there were 22 enrolled. And these women were randomized to receive either a probiotic intervention, so a probiotic sachet that they consumed twice daily, or a placebo. And they consumed that for 56 days. And we have saliva, CAPScan, and stool samples at their baseline, and then again at day 56, their last day of the intervention or placebo. And these were all pregnant women coming from areas at high risk for environmental enteropathy. So there was no inclusion criteria indicating that they had to have a biopsy or any proof that they had EE. So for us, this is a good population to kind of understand what we're, think a lot about how we can maybe define EE using this new device and the samples that we can collect. And so in this study, we have a new generation of the CAPScan device. So the outside looks quite the same. So this is what our study subjects get. They get a little packet and they open it, and it's a little foil, kind of push out with the device in it. And inside is actually a very long tube. So this is what it looks like. It's a silicone tube, food grade. There's kind of no FDA required approval parts. There's no electronics or anything in it. And this tube gets curled up inside that device, and then it slowly unfurls as people, as it's going down through the small intestine. And it takes somewhere, depending on an individual's peristalsis, somewhere from 3 to 5 hours to get through the entire small intestine. Once it gets into the stool, and this was the case with the Generation 1 device, but even more so with this device, it is a very, very small opening. It works via capillary action. So once sufficient moisture has been reabsorbed into the human host, like stool cannot get up into this. It can only collect fluid. And so basically, once it's past the ascending colon, where things are a little bit like viscous enough to liquid enough to get in, we can't really sample much past the ascending colon. And so in this study, instead of having subjects swallow 4 different devices, we had them swallow 2. One had a white cap and was designed, and the white cap is just this little piece so we can tell them apart when we retrieve them from the stool. This was the one that was designed to open higher in the small intestine and sample throughout the entire small intestine. The blue device was designed to open in the ileum and sample throughout the ileum and into the ascending colon. So we kind of have the devices overlap in the ileum, but we have really from the upper small intestine all the way through the ascending colon. And we, this slide is just to show you, we had individuals consume 2 at baseline, 2 at end line. So over 220 capsules were consumed. And almost all of those retrieved. Of course, with fieldwork, you always have funny stories, and I'm happy to share some of those later if there's time. One of the things that we're doing now is actually determining bioburden. So we've used a spike-in, a known microbe that we spike into the sample and use that to calculate the total bioburden. So we can calculate the bioburden at different areas along the small intestine. And what's interesting is that we already are seeing some very stark differences between our study sites. And we don't know if this is differences due to population level, differences in bioburden or biomass in these locations, or if it might be something to do with sample handling procedures. So for example, these 2 countries are the ones that got it onto cold chain earlier. And so we're following up with the field team currently to kind of understand the field dynamics and how those might relate to some of these results. But as much the same with our cohort from Stanford, we actually see a very nice and clear separation between the small intestine, the stool, and the saliva. But in these populations, we definitely see much more of the small intestine looking similar to stool. And one hypothesis of environmental enteropathy is that it's actually bacterial overgrowth into the small intestine. So colonic bacteria kind of overgrowing up into the ileum. And that would be consistent with these findings. But again, this is data analysis currently underway, and kind of more to come on that. We also have seen high prevalence of a lot of pathobionts. So I'm just grouping here at the general level. We're doing some more in-depth analyses at this point, looking at virulence genes, but that data is not complete yet. But there's a high prevalence of Escherichia and Klebsiella, and not only a high prevalence, but also actually high abundance. So we have some CAT scan devices that are predominantly, like almost 100%, either E. coli or Klebsiella. And we're also trying to, kind of suss out, is that overgrowth of these species during transit through, so transit through the small intestine is about 5 hours, but then we have to wait till it transits through the large intestine also, which takes, the total transit time from swallowing the device into retrieval in stool is about 32 hours on average. So again, trying to understand some of the dynamics, but it has been shown in duodenal biopsies from individuals with environmental enteropathy that there is overgrowth of Klebsiella and E. coli in the small intestine, and that often these are canonical pathogens, but they do produce some enterotoxins that, so we are now looking for those genes in this data. One of the really exciting things for me is that we've now started doing immunoprofiling of the small intestine as well, using a Luminex panel, 115 different cytokines, chemokines, and growth factors. And much is the same as we saw with the kind of the microbiome and the metabolites. We see a very, very big difference and kind of clear signal of which are the cytokines and the kind of a distinct immunoprofile between the small intestine and the stool. And so here the stool is the brown color, the green is the small intestine. And in this PCI, you can see that they very nicely separate. And so now I'm going to show you, this is within a single individual. And what I want to emphasize is that with this device, we can actually take multiple samples in the small intestine. So I told you that each subject swallowed 2 devices at baseline and 2 devices at endline. So this is one individual, and their white device is the first 3 points on the left, and then their blue device are the next 3 points, all the points in green, so on this upper left, and then the stool sample at baseline is that brown point. So that's the stool sample that the devices were collected in. So think of it as the farthest most left point is the highest in the small intestine, and you're just going incrementally down the small intestine. And you can see, for example, in granzyme A, at baseline, there are certain segments of the small intestine that we see elevated levels. And this is a log2 scale, so the move from 5 to 8, that is to like 6 times higher. And this is one of the 115 cytokines that we look at, but actually we see these unique, distinct spatial patterns in many of the cytokines. And when I was talking about the biomarkers earlier, I forgot to mention I had specifically named 2, myeloperoxidase and lipocalin-2. And we actually included those in our Luminex panel, and MPO is myeloperoxidase. And so this is one of the most commonly used fecal biomarkers for inflammation. And what we see in this subject is that the levels in the small intestine are orders of magnitude, higher than what we're seeing in the stool. And so is this really a good proxy? Is this really a good biomarker for what's happening in the small intestine? Are these fecal measures of inflammation relevant to environmental enteropathy? So this is one individual. You might say, oh, okay, that's just one person. But here are the correlations for myeloperoxidase on the left and lipocalin-2, which is another of the biomarkers that we use, fecal biomarkers that we use. Here are the correlations, which, as you can see, there are none, between what we see in the small intestine and what we see in the stool. And so this is really challenging, I think will be revolutionary for the field to understand that these biomarkers that we've been traditionally using might not be as relevant as we have expected. So in summary, the study of EE has been limited due to kind of all these challenges in studying the small intestine. But now we have new tools that we can passively sample humans during normal, normal digestion and normal, kind of daily life. And I hope I've convinced you that the small intestine ecosystem, both the microbes, the metabolites, and the immunoprofiles are quite different than what we see in feces, and that these classic EE fecal biomarkers might not be as relevant as we had thought. And really, we're at the beginning of this exciting new time where we can actually use these new tools and start to understand the condition, start to understand how interventions that we are targeting to improve EE are actually affecting the biology in the small intestine. So my lab is doing some work now with measuring IgA secretion and how the interventions are influencing IgA secretion and also IgA targeting of specific microbes. Yeah, so it's a really exciting time. I would like to say thank you to, projects like these are huge team efforts, and there were a lot of people involved, particularly the team at Stanford who helped with the first CAPScan study, and also our collaborators at UC Davis who did all the metabolomics work for that, and the team at In Vivo who has designed and manufactured these really cool devices and allowed us the opportunity to partner with them. And then to the Gates Foundation and all of our partners for the EMP work that I shared. And as I mentioned, that's ongoing work. We're in the middle of the second study now and really excited to see and learn from these populations. And a huge thank you to our study participants, because swallowing these devices and then collecting your feces for us for multiple days is not always a pleasant task, but people in general have been really great and supportive. And I've even spoken with some of our field participants in Bangladesh this summer. We were worried and they were like, yeah, it's not that big a deal. It's great, so we're thrilled that people are willing to participate in the studies. And with that, I'd be happy to take any questions.
Thank you so much, Dr. Grembi. This is so cool. I'm sorry I had to pop on a few minutes late. And Alyssa and I apologize to everyone that was here. I think we had basically one of the Zoom raiders pop into the chat, an external person. So we'll work with IT to make sure that doesn't happen again. But your presentation was great. This technology is so cool to me. It's so exciting. I think I got really excited when I first heard about like the cameras people can swallow, but it's the same issue that you were talking about, that those patients need to be fasted and cleared out and all of this to get any good data from that. So I think this is really interesting. I have a couple questions, but let me open it up to everyone else first. Does anyone else have questions for Dr. Grembi? Well, I'll ask mine then. So I'm always like, I don't do microbiome work. And so I'm always interested, like, I know there are individual differences, there's sex differences, there's regional differences. So how do you make sense of all of this data when you have, such a wide population, multiple geographic regions, multiple intestinal regions? How do you make sense of like what's normal or not?
Yeah, no, that's a great question. And in fact, that's one of the biggest challenges. We don't have any defined healthy people in our study. Like we need to define that. And so we've been doing kind of two things. So we have the immunoprofiles that I showed, the cytokine, chemokine panels, and we've been clustering those, and we kind of see like a Th1 profile. And so we're kind of looking for groups of biomarkers that might be moving in the same direction, and can we group people into that? And then we also have the traditional EE biomarkers, which are 4 fecal biomarkers and 6 systemic biomarkers. And in fact, when we cluster women using just those 10 biomarkers, we see a really interesting 3 groupings. One grouping is women who seem to have above average intestinal inflammation. So fecal, like, colonic inflammation. We see a group of women who appear to have above average systemic inflammation, and that's almost all of the systemic markers. And then we see a group of women who appear to like just be normal. They don't have inflammation, either systemic or in the colon. And so we're trying to understand, it'd be very easy to label the ones that have no inflammation as the healthy group. But given that this is a population with high exposure to enteric pathogens, we want to be really careful to make sure if they have no inflammation, is it that their immune systems have kind of said we give up and we're not, and so we're exploring a lot more. We actually have qPCR data on pathogens that we're trying to integrate into our analyses. But it's a really great question, and I don't think we have an answer yet. It's one of the things that we struggle with. Also, when you're thinking about the small intestine and you want to compare the small intestine, do you look at the highest level of inflammation anywhere in the small intestine? Because we have these up to six measures within an individual at a given time point. Or do you take the average? Because if you take the average, then you're basically dampening your signal. And so we are basically doing the analyses in multiple ways and trying to understand and kind of get a better sense. But we literally got this data back within the last few months, and we're kind of knee deep, and I really hope to be able to answer some of those questions in the coming months.
That's so cool. Do those those kind of three phenotypic groups, are you seeing any trends in like their clinical picture or like symptomatically? Is there any correlation?
So we're actually trying to understand the relationship between that and the small intestine cytokines, but also the kind of clinical outcomes we have are birth weight, preterm birth, and some of the child markers. And so the challenge, what I'm hesitant again to make any like definitive statements on that because is it that they have EE or is that their diet was undernourished? Like these women are not on a controlled diet. So if they're having children with low birth weights, or is it because they just weren't getting sufficient nutrition, or is it because and that is, I think, kind of convoluting. So I really think we need studies that are designed and capturing, either putting people on a defined diet or capturing that nutritional history really, really well. And I'm not sure that we are doing that yet for these studies, but I'm advocating for that in the ones moving forward for sure.
That's awesome. Well, thank you so much. This is innovative and at the forefront already. So I'm excited to see what more we could do. I'm thinking of thousands of studies that could be done with this kind of tech. So that's very cool.
Thank you.
And Dr. Grembi, thanks again for being here. It was awesome, super interesting. I learned a lot, and I look forward to talking about this with lots of people.
Yeah. Thank you very much.
Thank you all.